Welcome to Chapter 9, Part 2, The Tree of Life. Here are the objectives for this part of the chapter. As always, I expect you to have these mastered by the time we take our next exam, which is coming up soon. Make sure you take advantage of the recordings to help you hone in on these objectives. In this topic, we will talk about the Tree of Life and how evolution helped develop the Tree of Life. We will wrap up this chapter by examining the human genome. All of these concepts should be building on what we have talked about in the previous 3 chapters. Just in case you aren't sick enough of hearing about selective pressures, we're going to spend this whole slide discussing these pressures. We keep talking about how selective pressures helps encourage certain genes to be passed from one generation to the next. While this is true, the process is not neat and tidy. I may have a mutation that makes it likely that I will live to be 150 years old. But if I die in an accident, no matter how awesome my mutation was, it will not be passed on to the next generation. The converse is true as well. Someone may have a detrimental mutation, but as long as they achieve reproductive success, the mutation may be passed on. Even this is a simplified version of selective pressures. Don't forget that sexual reproduction also requires the genetic material of another individual. This could also alter the ability of the mutation to be passed on to the generation after next. Even if we pretend for a moment that this mysterious mutation is continued within a family tree, it is not likely that eventually the offspring of this family will become members of the X-Men. Most of the mutations that would be passed along are neutral mutations, meaning that while I may have an adenine in my sequence where you have a guanine, there will be no noticeable difference, and my offspring are no more likely to succeed than yours. The reason that our mutations are far more likely to be neutral than anything else goes back to how hard our cells work to maintain the genome. A mutation in any of our absolutely necessary genes would result in an incompatibility with life. This is why our ribosomes are so conserved. Changes to this sequence are far more likely to be detrimental by causing significant issues that may hinder development and embryogenesis, or enough in life that the person may not reach reproductive maturity. Highly successful mutations are hard to observe because, once again, it is highly unlikely that a baby is going to be born with any of the characteristics we associate with comic book characters. Instead, it is far more likely that the baby may be born with traits that make it far less likely to develop cancer or lack cellular receptors, the result of which leads to immunity to HIV. We observe this kind of traits within our population, but have yet to identify if these are genes that have been inherited or if they are the result of something else. Especially in cancer, it is difficult to track these because of the huge environmental exposures that families tend to share. We've gotten a little off topic, so let's get back to these variations in the Tree of Life. So while negative or positive variations are possible, most of our variations are neutral. While these mutations don't affect us, we are able to see these neutral mutations in the Tree of Life. You can see in this picture the percentage of nucleotide substitutions in primates. As you can see, the orangutan is one of the primates furthest separated from the rest of the tree. Their genome contains about 3% variation of nucleotide sequences compared to us, while the chimpanzee and us only have a variation of approximately 1.2%. If you remember that we have 25,000 genes, each with potentially thousands of nucleotides each, these differences can be thousands of nucleotides. Remember that some of the substitutions are neutral, while others are the changes that make us us and chimps and orangutans. Let's explore this comparison between humans and chimpanzees a little more. We are obviously different species. Physically, while we look more alike than compared to, say, a dolphin, we still are physically very different. We share almost all 25,000 genes with slight variations, and our chromosome structure is almost identical. We have 23 pairs of chromosomes, while chimps and other higher primates have 24 pairs. Just like in our deer example a few chapters ago, in our chromosome structure we have fused 2 chromosomes together to form our chromosome 2. while the other higher primates have two separate chromosomes but contain the same genes. Just like the deer, the arrangement of the genes on the chromosomes does not have a large impact on the reading and coding of the genes. What about all those mobile genetic elements we've been discussing? We know that there can be variation within our living population. This must create a large difference in chimps and humans, right? Actually, no. We have seen that nearly 99% of the elements that are in the same place in both of our genomes. So where does variation arise? The answer is literally here and there. On the slide, you can see 5 portions of the gene that codes for something known as leptin. Leptin is a hormone that helps regulate food intake and energy use. This is not the entire gene, just 5 sections of it. As you can see in each section, there are 5 single base substitutions. Anywhere that is not highlighted is assumed to be the standard sequence of our last common ancestor. Out of these 5 mutations, only one produces an amino acid substitution, and given that leptin is present in and functioning in both species, it can be stated that these mutations are neutral mutations. Before we move on, just to explain the gorilla reference in the image, these references are used to help determine which sequence the common ancestor may have had. Given the gorillas broke off earlier in the history, it is likely that the common ancestor had the sequence that exists in either the chimp or the human that agrees with the gorilla. Out of the 5 changes, 3 of the chimp changes were more than likely to have been present in the ancestor, and 2 of the changes in human were more than likely to have been present in the ancestor. Let's take a step further back and compare our genome to that of a mouse. Remember that compared to the chimp, we only have minimal changes throughout the genome, and often not resulting in amino acid substitutions, and even more rarely changing the gene product. Let's start with the same comparisons we did in the previous slide. As would be expected, our genomes and the 25,000 genes are pretty similar. We are both mammals with comparatively similar body plans, as opposed to that of a fish or a bird. After this, things start to show changes. Remember, in the chimp, 99% of our mobile elements were the same, and we have minimal differences in our chromosomal structure. However, in mice, not only are the mobile elements in different places, they aren't even the same elements. They are similar, but not the same like we saw in our comparison to chimps. Researchers have estimated that the rearrangements of these mobile elements and the genes within the chromosomes that are the result of these 180 breaks within the genome. While this may seem like a massive amount of changes, don't forget that we still share a lot of conserved regions or conserved synapse. Let's look at the leptin gene again. This is a shorter segment of the leptin gene than we looked at in the chimp, but even in the segment here, there is approximately 50% change between the 2 sequences. While it's easier to look at the highlighted segments, look at the segments that are not highlighted. These are likely to be sections that are where mutations occurred within these regions. The resultant organism was not able to survive. This is referred to as purifying selection and shows us where genes cannot be changed no matter what. When these segments are identified in prokaryotic and eukaryotic cells, we know that changes in these regions are almost immediately fatal to the cell. Let's take this discussion one step further to talk about comparative genomics. This picture shows the comparison of a short segment of DNA in one gene. This picture gets a little confusing, so let me explain how to read it before we talk about what it means. At the top of the picture is the map of the gene where the red bars indicate exons. Then we zoom in to 10,000 base pairs with one exon that is still in red, and the segments in blue are conserved across multiple species. Below this map are a lot of tiny bar graphs for each species listed. The 10,000 base pair segment is broken down into 100 base pair units. You can see those divisions in gray at the bottom in chicken and fish. The green is then a bar graph that indicates how conserved the segment is. Only regions that fall between 100 and 50% are noted. So if we look at the fish, we can see that there are about 3 areas that are similar to our gene, while in the chimp, most of the gene is identical. What we can see by doing this analysis in an exon that is highly conserved across many different species, even across different types of animals. However, as you can tell, the introns start to vary greatly the more distant the species become. We see a large fall off when we jump from mammals to birds and fish. When we compare these segments across multiple genes, we can see about 5% of our total genome matches most mammals, and of these only about 1/3 code for actual protein products. Others we have identified as producing RNAs or in regulatory sequences, and others that we have no idea. In something that is conserved across species, it is likely that it is very important, and the fact that we don't know highlights just how much we still don't understand about cellular biology. When we compare mammals to fish or birds, the dynamic differences make direct comparison very muddy. What jumps out very quickly are the areas that are the result of purifying selection. These help highlight, even more than in mammals, which genomic sequences are absolutely required for life. On the macro scale, we can see that most mammals' genomes are of a similar size to ours, while reptiles, fish, and chickens vary dramatically. This is likely the result of gene and whole genome duplication that we discussed earlier. One example is of the pufferfish. The fish's genome is about 1/10 the size of ours and contains minimal introns. However, the introns they do contain are very similar to ours, indicating that these introns are very important. When we compare gene sequences between species and start to build the tree of life, we can see that organisms can quickly add and lose genetic information. Those with minimal genomes, such as the pufferfish, are examples of what a higher ordered animal must have to survive. So how does all this information lead to the tree of life? We use the same methods we have been discussing to look for regions of the genome that are part of the basic building blocks of life. One of these is the gene that codes for the rRNA found in the small subunit. As you can see on this slide, there's a lot of conservation, highlighted in green, between humans, archaea, and bacteria. Where the differences are, we can compile the information to help us learn where and how we diverged. We are also able to use this information to reconstruct an idea of what the common ancestor's genome must have looked like. Remember that while we were talking about all this in terms of genomics and sequencing, we haven't been able to use this data forever. Darwin and earlier scientists constructed the original trees of life based on observation. It wasn't until 2001 that we were even able to sequence the human genome. If we have been able to determine this much information in this short amount of time, imagine what the future holds for the development of the tree of life. Let's wrap up this chapter by examining the human genome. To do this, we are going to discuss how our genes are arranged, what makes us human, what variations are present within our population, and finish up by discussing briefly what we still don't know. We've mentioned it before, but the human genome is the entirety of our genetic information. Each cell in our body contains the entire sequence, and it is only through gene expression that cell differentiation arises. Our entire genome was first sequenced in 2001 and essentially rewrote this textbook with the information gathered from the sequencing and subsequent experiments. And while we may have the sequence, we still don't understand how much of our sequence works. Let's talk about what we do know. We have talked about this a lot, so hopefully this is a refresher. On this slide is a data table we have seen before showing some statistics about our genome, as well as a map showing the components of our genome broken out by percentages. Remember that what we learned when we sequenced the genome is that our genome contains genes, mobile elements, and junk DNA. Almost half of our genome is made up of these mobile elements, which surprised researchers. When the genome was sequenced, we learned that genes do not simply code for gene products. They instead hold both introns and exons, as well as regulatory sequences. Of the genome sequence that we know, that 5% is highly conserved across mammals, which means that it is likely that these sequences are required for life to occur. The final conclusion on the arrangement of our DNA is that it is really just a mess. A lot of it is highly individualistic. This partially explains why we have the basics understood about the arrangement of our genome, but we still don't understand a lot of it. So when all this information makes us human and not, say, a chicken, dog, or chimp? The answer is that we aren't sure. Once both the genome of the human and the chimp were completed, researchers set to finding the differences between us in hopes of identifying the genes that make us look so starkly different. They set about this by looking for areas that have conserved sequences, such as the ones we saw earlier. What they found within these regions are some pretty big shifts between humans and chimps compared to another animal. For example, one gene was found to have 18 differences between humans and chimps, while the same gene between humans and chickens only varied by 2 nucleotides. If you look back to our comparison between species before this, this is a startling discovery, given that mammals traditionally are more similar. What this suggests is that after we split from chimps way back, our genome underwent some accelerated changes in a few but significant regions, resulting in the shifts that we do not completely understand. So what does this all mean? We aren't entirely sure, but it sure is exciting. What we have determined with a lot more detail is what makes us different within our own population. As you know, no two people are alike. Even identical twins have epigenetic differences. What makes our genome so different yet so similar? We all vary by about 0.1%. Remember that we vary from chimps by about 1.2%. This variation adds up to about 3 million different genetic differences in each set of chromosomes, which is a heck of a lot. Not all of the variation is recent either. Evidence suggests that a lot of our variation came from when the human population was very small, about 10,000 years ago. We have 3 main variations that we note occurred comparatively recently. The first of these single nucleotide polymorphisms, or SNPs. These are areas in our genomes that different populations vary from each other. Given our history, it makes sense that people from different regions would have similar sequences, while others show a difference. For example, on the segment of the human population that may have an AT base pair, another group may have a GC base pair, and 2 random people chosen from the world population would differ from each other by about 2.5 times 10 to the 6 SNPs scattered throughout their genomes. SNPs are very useful in genetic studies, and we talk about them in more detail in Chapter 19. You can see an example of an SNP analysis on this slide. The second form a variation comes from duplications and deletions. We have 100 long blocks of DNA that differ from person to person. We have identified some of these, but others we still aren't sure of. Even more so, we don't know entirely how these variations contribute to the human genome and what they mean in terms of our evolution. The last point of variation that we are aware of comes from areas of repeated patterns of base pairs. These regions are particularly open to DNA mutation. While we still don't quite understand the role this variation plays on our genome, we have been able to harness this variation through DNA fingerprinting that allows us to match genetics to either other family members or to samples of DNA left at crime scenes. While once again we don't understand all the roles these variations play within our population, or know how the world becomes more and more globally connected, how our genome may evolve, we do know that more of these variations are silent, and more do not explain why we look so different from each other. We will discuss this further in Chapter 19 when we talk about what we know about our physical traits and the genome. So, what do we still not know? A heck of a lot. In the coming years, hopefully most of this will be revealed, but our current lack of knowledge is not due to people not working on these projects. It is instead due to the complexity of our systems. Not only does the sequence of genome contain regions that we aren't sure about, the epigenetic markers can alter how this process is read. And even after that, the gene expression of these products can be varied in so many steps. One area that muddies the water is the alternative gene splicing we discussed before. Each gene can be cut in a variety of ways. That makes these products difficult to track back to the gene. Another aspect of gene control are miRNAs. We only just discovered these regulatory sequences. With this additional layer of product control, we can also determine what proteins are being copied for where. So what do we still not know? A whole heck of a lot. Well, this is the end of Chapter 9. As always, please review the objectives list and let me know what questions you have when we come to class. If you'd like to discuss any of these objectives one-on-one, please let me know.